For decades, we figured huge parts of our DNA were just evolutionary leftovers, useless junk. But what if I told you that a sleeping giant lives inside that so-called junk, and when it wakes up, it can unleash one of the most feared diseases of our time, cancer? So let's just start with a number that is, well, it's kind of mind-blowing. 50%. Imagine if half of the entire instruction manual for building you was just dismissed as junk. For the longest time, that's exactly how science viewed a massive, massive portion of our DNA. And this isn't just, filler material. This 50% is packed with millions of tiny, mysterious sequences called transposable elements, or TEs for short. You can think of them as jumping genes that have this incredible and kind of scary ability to copy and paste themselves all throughout our genetic code. Okay, so you're probably thinking, wait a minute. If half my DNA is this wild rogue code that's jumping all over the place, why aren't our bodies just a complete genetic mess? Well, the answer is that our cells have evolved these incredibly sophisticated guardians to keep these jumpers on a very short leash. But before we meet those guardians, let's just be crystal clear on what we're dealing with. These transposable elements, they're ancient. I mean, some are literally the remnants of old viruses that wormed their way into our ancestors' DNA millions of years ago. They are a deep, fundamental part of our evolutionary story. So, how do our healthy cells keep these ancient jumpers quiet? Well, they use this brilliant process called DNA methylation. You can picture it like putting tiny chemical locks all over the TEs, physically holding them down so they can't move and they can't make copies of themselves. Think about your genome as a perfectly organized library, right? These epigenetic locks are the librarians, making sure all the books, our genes, stay on the right shelves. This keeps our genetic instructions crystal clear and prevents everything from getting scrambled. But, and this is the crucial part of the story, in the development of cancer, something goes terribly, terribly wrong. This elegant, tightly controlled system starts to break down. Here's the breach. In a cancer cell, a process called hypomethylation starts to remove those chemical locks. And the result? The sleeping giant awakens. The TEs are suddenly free to start jumping all over the genome again. So the difference is just night and day. In a healthy cell, everything is orderly. The TEs are silenced, the genome is stable, the epigenetic locks are firmly on. But in a cancer cell, it's the total opposite. The locks are off, the TEs are active, and the genome becomes dangerously, catastrophically unstable. So what actually happens when tens of thousands of these TEs are unleashed inside a cell? The result is pure genetic chaos, and it causes damage in three major ways. First, and this is the most straightforward one, they can cause a ton of damage just by where they land. A TE can jump right into the middle of a really important gene, basically scrambling the blueprint for how a cell is supposed to work. When one of these TEs lands smack in the middle of a gene, it's like a huge disruptive typo in a critical sentence. It can lead to a broken protein, or it can shut that gene down completely. Now imagine that happening to a gene that's supposed to protect you from tumors. Yeah, the results can be devastating. Second, their activity doesn't just mess with single genes. It can physically break our DNA. We're talking about massive, large-scale damage to our chromosomes. This creates what scientists call genomic instability, which basically means the whole genetic blueprint starts to shatter. Entire chunks of DNA can be deleted or copied in the wrong place or moved somewhere else entirely. This is the exact kind of chaos that lets a cancer cell grow and get more and more dangerous. And finally, there's the third path, which is maybe the most direct route to cancer. These TEs can land near genes that are known to drive cancer growth and flip them on like a light switch that gets stuck in the on position. And look, this isn't just some abstract theory. We see this happening in actual patients. In some bladder cancers, a TSO is what switches on a cancer-causing gene called MET. In melanoma, it's a gene called ALK. In lymphoma, ERB4. In every one of these cases, a jumping gene is the culprit, flipping a switch that tells the cell to grow uncontrollably. So, what does this all mean for how we understand cancer? Well, it gives us a fascinating and, frankly, kind of scary new piece of the puzzle. So let's put it all together. It's like a chain reaction, right? It can start with stress on a cell, from things like inflammation or even radiation. That stress causes the epigenetic locks to fail. That awakens the TEs. And once they're active, they switch on cancer genes and destabilize the whole genome, pushing that cell down the dark path to becoming cancerous. And What's so important to get here is that this isn't some rare, bizarre phenomenon. Scientists are now realizing that the chaos caused by these jumping genes is a fundamental part of the story for many, many different kinds of cancer.
And this just completely flips our old understanding on its head. That vast, ignored portion of our genome, it's not junk. Not at all. It's now a critical area for research, a prime suspect in what causes cancer, and maybe, just maybe, a whole new frontier for developing treatments.
And that really leaves us with a final, pretty provocative thought. If we've misunderstood almost half of our own genetic code for this long, if we've been sitting on a sleeping giant this whole time, what other profound secrets are still hiding in our DNA, just waiting for us to find them?